talk today. It really means a lot that y'all showed up to hear about my research. It's been a long time coming, 7 years of grueling work, but also very exciting work as well. So you can see my title is Molecular Systematics of Porroglossum from the family Orchidaceae. We're going to be looking at phylogenetics, floral snaptrap kinematics, and fragrance. Okay, so I'm just going to give a brief overview about orchids and then kind of work my way down to Porroglossum, and then I'll have a short intro for each chapter as well to kind of orient you about what we're talking about. Okay, so the big overall overview that we have here, the family Orchidaceae is composed of 5 subfamilies, as you can see here. The largest of them is Epidendroideae, which is most important for today because that's where subtribe Porroglossum is found. Right? So the subtribe Porroglossum is the largest subtribe of Orchidaceae, as you can see here. There's more than 4,000 species and 29 genera. Alfonso, myself, and Ken have also written a paper where we're condensing that down to, I believe, about 8 genera. Hasn't been accepted yet, but it's in review right now, so there may be some taxonomic changes there. But it contains about 15 to 20% of orchid diversity. I guess one of the most intriguing things about it is even though it contains so many species, 4,000 species, you can see that the Porroglossum lineage arose very early on in terms of geologic time, between 14.2 and 21 million years ago. So it's had a lot of time to speciate, it's speciated a lot in a very short amount of time. Right? And so for Porroglossum, or what a lot of people call pleurothalids, a lot of people that are really interested in them are horticulturalists, and they usually are into these tiny obscure flowers from genera such as Stelis. However, there are also really amazing ones that are a lot larger and bigger showy pieces, such as from the genus Masdevallia, which happens to be the most close relative to Porroglossum, which is going to be the topic of today, like I talked about earlier. Right? So this is just a paper from Pridgen et al. In 2001, where they used nuclear ribosomal ITS and I think some plastid genes as well. But here they were just trying to determine the subgeneric ranks within, the generic ranks within the subtribe. So essentially trying to figure out what the genera are in the group. And they found that Porroglossum was monophyletic and sister to Masdevallia, as I was saying earlier, its most close relative. So here's a representative of Porroglossum. Right here, the flowers are a few millimeters, not longer than your fingertip. They're really small, even though they look big here from being zoomed in. So the orchigenus Porroglossum has 54 described species that are distributed throughout cloud forests in the Andes. And they're distributed from Venezuela south to Colombia, Ecuador, Peru, and Bolivia, with a high concentration in Ecuador. Most of the species are found in Ecuador, endemic there, or they may have just not been found in other countries as well, being that they're extremely rare. But another intriguing thing about Porroglossum is that it's also a really young lineage. It arose between around 3 and 4 million years ago, according to Givnish et al., and also a paper by Perez Escobar. Right, so a really young lineage with a lot of species, which can be problematic, which I'll get to in a little bit. So this is just a plate of flowers from Porroglossum, so different species. I just wanted to highlight the tremendous floral diversity that you see here. You can see that we have tons of different colors, forms, sepals that are thicker, thinner, and longer. We have peduncles that are hairy, peduncle meaning a flowering stem. We have peduncles that are smooth. This species here is Porroglossum edwardi. It's the only one that has a red flower, and it's also got a really weird growth habit. Kind of grows along the substrate with its rhizomes. And then we also have some that kind of have bumpy sepals, as you can see. So a lot of floral morphology, a lot of diverse floral morphology there. So the thing that makes Porroglossum the most unique, at least in my eyes, is the fact that it has an active snap trap. So the flower is modified into a pollination trap, where the labellum, as you can see here from this line drawing, when an insect lands on it or a mechanical stimulus is applied to it, it will snap shut. It'll either snap shut rapidly or it'll take a while to snap shut, as you'll see in my research later on. But here you can see it in the open position and the closed position for Porroglossum. There are 3 other lineages within the Porroglossum that also have an active snap trap. Acacia, Condyligo, and one species of Masdevallia. However, none of them have differentiated as much as Porroglossum. Again, Porroglossum has 54 species, and other ones such as Condyligo and Acacia only have a few species. Okay, so these are my 4 dissertation chapters. I'm going to try not to just read off the screen and kind of tell you what I'm going to talk about in each chapter. So chapter 1 is, like Ken was saying, I used a traditional Sanger sequencing approach to try to figure out the phylogenetic history of the group Porroglossum. And I did a traditional approach where I used an ILD test to determine if the genes were congruent and if they should be concatenated or not. And then I looked at individual gene trees as well with that approach. And then I also did a concordance approach with Bucky. I know I didn't have that many genes to do a concordance analysis, but I wanted to go ahead and cover all of my bases anyway, where I had the chloroplast DNA as 1 gene versus 2 other nuclear genes, which I'll get into in a little bit. Chapter 2 is my genotyping by sequencing chapter, where I extracted the whole genome of over 30 species and then sent them off to the biotech lab where they cut it up with a restriction enzyme and essentially made very small loci that you can look for SNPs or informative positions in order to create a phylogeny. And I also utilized the trees that I got from that using Stax and Pyred, the 2 main pipelines, with morphometrics to see if there was any, any evidence of any evidence of the groups separating in terms of the floral morphology. Floral morphology versus the molecular data. Then for chapter 3, this is probably my most interesting chapter, my most favorite chapter as well. It's my kinematics chapter. Kinematics just meaning it's a branch of physics that just looks at motion as a series of data points. Right, so if you have a video with something moving, you can put a pointer on it and continue to track it through time and calculate things such as velocity, acceleration. So looking at the snap trap and how it acts in different species. And then my last chapter, which I must admit is very thin at the moment, however it does have some very interesting data, is the characterization of volatile organic compounds. So essentially collecting floral scent from 10 of the species of Porroglossum. And all of this work was done in Gualaceo, Ecuador, in the greenhouses of Equigenera. Or in some cases, I actually had the plants imported where I had purchased them, and I modified a wine cooler so that I could grow them here long enough to be able to get vouchers and also extract DNA from them for the phylogenies. All right, so chapter 1, introduction. Just really quick, Porroglossum taxonomy. There's only been 2 taxonomic circumscriptions that have been written. One by Lure in 1987 and another one by Auburn in 2004-2005. The first one had 25 species and the second one had 34 species. So as you can see here, Lure had 2 subgenera, Edwardia and Porroglossum, with Edwardia having that single species P. Edwardii, the red one that I showed you earlier. And then subgenus Porroglossum was split into 3 sections, Porroglossum, Echidna, and Torte. And I'll get into the morphological characters in a minute that they utilize there. And then here, you can just see that Auburn elevated Echidna from the section status up to the subgenus status, and he also added section Torte and subsection Mordax. So essentially just shuffling around Lure's taxonomic circumscription a little bit, but also classifying a new group, which you'll see will come into play later, subsection Brevicephala, which hadn't been described by Lure. Right, so the morphological characters that were utilized for all of these groups, for subsection Brevicephala, the sepals have 3 raised veins, as you can see here, 1, 2, 3, that run along the length, and then the dorsal sepal is enlarged at the base, and then you can also see the tail of the dorsal sepal bends backwards. Sometimes it'll have a really long tail, sometimes it'll have a little bit of a nub, but the tail will always bend backwards. So those are the characteristics that were utilized to describe Brevicephala. Subgenus Edwardia, I've already talked about a little bit. It's characterized by the monotypic species Porroglossum edwardii. It has a long rampant rhizome, so it has that creeping growth habit. No other species exhibit this growth habit. And its racemes are also distantly flowered, as you can see here. Subgenus Porroglossum section echidnae is really easy to diagnose based on the pubescent peduncles or hairy flowering stems. the only group that exhibits that characteristic. Section Porroglossum is characterized by resupinate flowers, meaning that they're turned right side up, and then the ovaries being more or less not twisted, as well as smooth or glabrous flowering stems. And this is where it gets a little bit tricky. Section Torte, which is described differently than section Porroglossum, is only differentiated by whether the ovaries are twisted and if the flower is non-resupinate or turned upside down. However, based on several monograph pictures and line drawings, I've seen species from other groups such as the Brevicipilla group that also exhibit the non-resupination character. So this may be a character that's not a great synapomorphy in this case. But however, this is how it's traditionally described. All right, so research questions for chapter 1. Number 1, is the orchid genus Porroglossum monophyletic, or do they all come from one common ancestor? Number 2, can traditional standard sequencing markers resolve interspecific relationships or at the species level, as well as deep internal nodes? By that I mean looking back through time, the backbone of the phylogeny, for lack of a better word, and this extremely young orchid lineage, remember it being 3 to 4 million years old. Does the molecular data set support previous taxonomic classification systems based on lure that I showed you a little while ago? And then, is there gene tree discordance that may suggest underlying evidence of biological processes such as hybridization, introgression, horizontal gene transfer, etc.? Alright, so chapter 1, the materials and methods. Just have one clean screen here for the methodology for taxon sampling. I collected fresh leaf material from all of these from plants that was growing in the wine coolers. So I had 40 total pleurothalates for the standard sequencing chapter. 28 of the 54 were from the species Porroglossum, and then you can see the rest of the outgroups here. The most distantly related one being Tricetella. That's what we utilize to group the entire tree, or multiple trees, I should say. For gene sampling, we had 5 genetic markers: nuclear ribosomal ITS, a low copy nuclear gene XDH, xanthine dehydrogenase, and then 3 plasmid markers: METK, RPL-L, and YCF1, which were utilized heavily in other papers and considered to have a lot of phylogenetically informative characters for not only orchids but other families of plants. So that's how we came up with this sampling for the plastid genes. All right, so once I got all of the raw data back from the Biotech Center, I utilized Genius to build contigs. Right, so essentially editing the sequences, making sure that the chromatograms match up and that the base pairs are good manually. Then I built the consensus sequences and aligned everything in muscle so that I had the alignment for each gene. Then I used the program PALP to run that ILD test. So I ran the ILD test many times. I ran it with the chloroplast data by itself. I ran it with the chloroplast data plus 1 nuclear gene, the chloroplast data plus a nuclear gene, and nuclear ribosomal DNA, essentially all the combinations to see if anything was congruent or not. And the only thing that I found was congruent was the chloroplast DNA. Everything else was treated as its own separate gene tree. Then I also used the J model test. I calculated likelihood scores for that for the combined nuclear and combined chloroplast data to determine the best models of nucleotide substitution to utilize for standard sequencing to build the trees. Then, of course, I used Cypress to build the trees. I used the maximum likelihood approach as well as a Bayesian approach. And the trees that I got, the resulting tree, individual gene trees that I got from the Bayesian approach, I also utilized for the concordance analysis to run Bucky. So running all of those trees at the same time to see which ones support certain relationships through concordance factors. Okay, so chapter 1, the results. This is just a table of all of the information that I got from the standard sequencing. So I guess a few things that I can highlight is, for instance, MATK, even though it has a lot of base pairs, you can see that it has a very low amount of variation or informative sites, being that it's a marker that's a gene marker, I should say, whereas other ones that are spacers have higher, such as RPL32 Turnell, which is a spacer, has a lot higher informative sites. And then also you can see here the models of evolution that were utilized, as well as the likelihood trees. So the lower scores would represent a better likelihood score. So just essentially showing you that I covered all of my bases here to build the phylogenetic trees. Right? So this is my first figure for the standard sequencing. I have 3 different trees here. This tree is the tree with all the chloroplast data, so RPO 32, YCF1, and MATK. This tree right here is the ITS tree, and then this tree is the XDH tree. The XDH tree is the most unresolved tree, as you can see. We have a lot of branches that are collapsed. However, we do have some groups starting to emerge, so these are species of the Brevicipilla group. For the ITS tree, we have a little bit more resolution as well, so we have the Brevicipilla group plus the Porroglossum group coming out with high support. But the chloroplast DNA tree by far was the best tree. Here we can see that we have support for a relationship between the Porroglossum group being sister to the Echidna group, those species with the hairy peduncles, and then the ones that were smooth and glabrous that either had ovaries that were twisted or not twisted, and then that clade is sister to the Brevicipilla group, the ones where the dorsal sepal tails were bending backwards. And you also see that this has a phylogenetic relationship in terms of the snap traps as well later on. And then all of these down here are just the outgroups that I utilize. I guess one other thing I should note as well, first thing I should have noted is that we also recovered Porroglossum as a monophyletic group with high support, 95% bootstrap support there. Okay, so this is the Bucky primary concordance tree. So we pretty much get the same relationship in terms of the clades that we get. We get the Porroglossum clade, the Echidnae clade, and the Brevicipilla clade. However, here the internal nodes switch. So instead of the Porroglossum group being sister to the Echidnae group, we have Porroglossum and Brevicipilla Brevicipilla and Echidna sister to Porroglossum. And then on the side here, I just have all of the taxonomic groups that were described by Luer and Auburn, just to show you that there are some taxonomic relationships that are supported, such as these species from what's would now be known as the Brevicipilla group. However, there are a lot of species that are non-monophyletic as well. So there's a lot of work that needs to be done in terms of the taxonomic groups when you compare it to the molecular data. Okay, so figure 3 here is just a plate of 2 potential hybrids. I realize obviously I can't call these true hybrids yet without more work, such as doing cloning and different things like that. However, based on the combined chloroplast tree versus the nuclear and ribosomal DNA, we see Porroglossum surgioid and Porroglossum TDI bounce around on the tree between 2 different clades, which is evidence that it could be hybridization or it could be another type of biological process such as incomplete lineage sorting. But there is other lines of evidence that it could be hybridization as well. As you can see here, the 2 potential parents, Porroglossum nudibara and Muscosum, are really similar, with surgioid being kind of intermediate between it. And then all 3 of these species also occur in the same department of Antioquia. Antioquia of Colombia. And then TBI as well looks kind of intermediate a little bit between Amethystinum and Porroglossum muscosum, and they all 3 occur in the province of Pichincha in Ecuador. But there's still further work to be done there to see if these are hybrids or if there's something else going on biologically with those species. Okay, so chapter 1 conclusions. Monophyly of the genus Porroglossum is highly supported, as I showed you by the chloroplast DNA tree. All of the 3 monophyletic groups were recovered: Porroglossum, Echidnae, and Brevicipilla. The interspecific relationships remain poorly resolved or unresolved, as you saw by a lot of those collapsed branches. Coalescent or concordance analysis approach, or in other words, utilizing Bucky, shows a conflicting tree topology when looking at the chloroplast DNA. So having the backbone of the phylogeny showing a different relationship. Within Porroglossum, preliminary molecular evidence partially supports Luer's taxonomy. However, we have some non-monophyles, such as the section Porroglossum. So section Torte is nested within section Porroglossum, right? So the ones that were where the flower was upside down and the ovaries were twisted. Those all nest with the ones that have the flowers right side up and not twisted. They form a clade. And then a subset of the species from the Porroglossum group form a clade that's sister to Edwardi, the one with the red flower. So a lot of work still needs to be done with the taxonomic group, like I was saying earlier. So some taxonomic circumscriptions. And then geographic and molecular evidence at least provide support for two potential cases of hybridization within Porroglossum, being Sergioid and TEI. But more work needs to be done to tease that apart. So then chapter 2 introduction. Some of these take a little bit to load the next slide. There we go. Okay, so I will admit I don't have a lot of slides for introduction for chapter 2, so I'm gonna just kind of talk about it logically. So in the past, reduced representation sequencing, which is known as RAD sequencing, restriction-associated digestion, is essentially an umbrella term for all of these types of techniques, such as genotyping by sequencing, easy RAD, which is just another version of RAD sequencing, double digest RAD sequencing, where you use 2 different enzymes. So essentially, it depends on how many enzymes you're going to utilize, how many steps you have. Are you going to have a sonicator step where you essentially shear all of those reads down to the same amount of base pairs, or in another case, GBS, where you're going to have reads that are going to be of different lengths. So a lot of different types of methodology that can be utilized here, but will give you different results in the end when you're going through the pipeline, or it may not be more difficult to interpret through the pipelines. However, all of these methodologies in the past were utilized for population genetics type work. It was the gold standard for that. However, recently, within the last 10 years, there's been more than a dozen papers that have been published on not only plants but animals as well, showing the phylogenetic utility of genotyping by sequencing at the species level. So there was once a thought that it didn't have enough resolving power to work at the species level. However, there's been multiple studies that have been published by Andrew Hipp and Darren Eaton, et cetera, that have showed that genotyping by sequencing can be utilized for species level recognition, especially for lineages that are really young in age, right? So this would be a good candidate for genotyping by sequencing. Okay, so like I was saying, here's just 2 studies that utilize genotyping by sequencing at the species level. So Eaton and Reed utilize it in Pedicularis to uncover relationships within a species complex that had been unresolved previously through standard sequencing. And the same here with Wagner et al., where they looked at Lake Victoria cichlids and were able to differentiate species that they had not been able to previously with standard sequencing. So the resolving power of genotyping by sequencing has been shown to be far greater than standard sequencing because we have a greater amount of DNA. However, there are some issues such as sequencing error and paralogs and different things like that that you have to look out for, which makes things like parameter testing or having a reference genome really key for these types of analyses, because you're essentially looking at anonymous loci. You're not going to be sure if the locus is from a gene or a spacer region or anything like that, essentially. So the research questions for chapter 2 is extensive testing of core parameter values necessary to assemble phylogenetically informative loci de novo with GBS pipelines. The 2 pipelines I'm using here are PyRad and STAX. How informative are GBS loci in terms of resolving relationships among interspecific taxa as well as deeper in time, right? So like I showed you earlier where we had the backbone and the phylogeny switching between different methods and then also tips collapsing. So would GBS be a better way of resolving those issues? And then when evaluated in the context of phylogenetics, can floromorphometric traits be utilized to elucidate taxonomic relationships within the genus? Okay, so chapter 2, materials and methods. Here for my sample size, I had fresh leaf material again for 43 pleurothalids. So I increased my taxon sampling to 37 species. I only used 3 species of Dracula and then Cranzanella amphracta to root all of the trees. Once I extracted the whole genome from all of these and quantified the amount of DNA in them, I sent it off to the UW-Biotech Center, where they essentially use a restriction enzyme to cut up the DNA, add ligand adapters to them, and then sequence 100 base pair reads that were then sent as a raw folder with barcodes attached to them. Then I use the Pyrad and Stacks pipeline to demultiplex and remove barcodes, so essentially sort all the samples to the correct species and remove all the barcodes from them. And then I generated genomic data sets consisting of de novo assembled loci. And I also utilized a method called parameter testing through DNA replicates, which was first described in the Mistretti-Yanes et al. Paper, which DNA replicate is essentially if you have a well of DNA samples that are being sent off for sequencing, if you put two samples that are exactly the same sample in two different wells, they're going to have different barcodes. So you're looking for the number of mismatches between them to calculate low error rates for things such as loci, SNPs, alleles, right? And then for morphometrics, I analyzed all of the floral shapes, labellum shapes, and petal shapes from all of the literature that I could find, such as monographs or any books that had pictures of the flowers in the labella, using the package Momox and R. And then I also used the L1OU package to see if I could detect any evolutionary shifts within the tree in terms of morphometrics. And I used the PBIC approach because it's considered the most conservative approach. The other approaches can give you a lot more shifts. However, the paper described that a lot of those shifts can be false or pseudo shifts. So just to be on the safe side, I used the most conservative one, even though it gave me a lot lower shifts than looking at the other ones. Right? So the reason why I have these two papers highlighted again are for the Stacks pipeline, I followed the Mistretti-Yanes et al. Paper for the parameter testing, and for the Pyrad pipeline, I used the Anderson et al. Paper for parameter testing. So I'm not going to go through each of these parameters one by one, but I just wanted you to see that these are the five core parameters within Stacks, and I tested each one extensively. By testing them, all I did was I took 10 species, I got replicates for each of those, so I had a data set with 20 species or 10 species, 10 replicates, and then I set each value, I changed each value while leaving the other ones the same. So for instance, if I was testing the value of 15 here, then all of these would have been left the default value, just as they described in the paper, and I did that for all of those values to determine which values give you the most amount of loci and SNPs, but also the lowest error rates in terms of loci, alleles, and SNPs as well. So trying to get low error rates, but also a high amount of informative data. Then I did the same thing for the Pyrad pipeline, right? So here are all the parameters as well here, and you can see that I extensively tested all of those parameters. And you'll see which parameters had more effects than others as I start going through the results here right now. So chapter 2, the results. I know these are kind of tiny, but I don't really want to dwell on them anyway. I just wanted to show you that for the Stacks pipeline, the M parameter and the N parameter seem to have the biggest effect in terms of the number of loci and SNPs recovered, whereas the big M parameter and the max locus Stacks parameter seem to have little or no effect. However, just because it showed in our data set that it was little to no effect, it could still have an effect on other data sets. It's essentially a per data set basis. If you look at the Mistretti-Yanes et al. Paper, you'll see a slightly different correlation than we have here. However, it makes sense that the M parameter would decrease the number of SNPs and loci as you increase it, because that's the the number of bases to make a statistical base call, and then these are just the error rates right here as well, looking at loci, SNPs, and alleles. So essentially, trying to find a sweet spot where we have a low error rate and a high number of SNPs and alleles. But it's also been described in the papers by Mistretti-Yanes and Anderson as kind of being subjective. So not only trying to find the optimal parameters, but also testing parameters around those optimal parameters to see if you get a different tree topology. Okay. So as you can see here, this was the most optimal data set that I had based on the phylogeny for Stacks. However, I tested all of these as well, and for the most part found the same topology. I'll talk about the few differences that I did find. However, I didn't test the parameters extensively here. So this is figure 3 from the snacks pipeline. As you can see, this is the Porroglossum group, which is composed of the ones that are recipient and non-recipient, particularly the non-recipient ones being up here, but they all form a monophyletic group with 100% support there. The echidna group, the ones with the hairy peduncles, also have 100% bootstrap support for the entire group as well. And then we also can see here that the internal node shows the Porroglossum group being sister to the echidna group. And then we have the Brevicipilla group here, which is monophyletic for these species. However, Atrix and Rodrigoi come out sister to the rest of the group here. Although Atrix and Rodrigoi tends to bounce around a lot in the tree based on the parameters that we set. So there could be other biological processes going on there that need to be teased out further. However, all of these species conform to the same or similar morphology as those two species. And then Porroglossum edwardi, at least in the stacks phylogeny, comes out sister to the rest of the genus. The one with the red flowers and the weird growth form. All right, so the pyrite parameter values, I'm not going to talk a whole lot about these as well. I'm just showing you that D, C, and MR show the greatest amount of change when you change the parameter values. And it makes sense for these parameter values to change as much as well because they're essentially filtering processes. So determining the statistical number to make a base call, or how many sequence similarities you have to cluster a sequence together. So the higher of a threshold that you make, the harder it's going to be to make loci SNPs. So essentially you're going to have a lot of allelic dropout, or a lot of SNPs, alleles, and loci dropping out of the data set. So the same trend of trying to find a sweet spot with the best parameters, but also testing a lot of parameters around there. So you can see here that I tested 7 data sets in the case of pyrite, and I used the 25%, 50%, and 75% threshold for all of those. So essentially, how many taxa do I need in order to make a locus? So if I have 25% of the taxa that have this locus, then it's included in the data set. If it doesn't have 25%, then it would be excluded, et cetera, et cetera. So this was our best, most optimal tree, which I'm going to show now. So for this tree, it has essentially the same relationship that we saw with the stacks tree, where we have a monophyletic Porroglossum group, monophyletic echidna group, which are sister to one another, monophyletic Brevicipilla group, which we now actually see Porroglossum, Atrix, and Rodrigoi nested within that clade with high support. And then Porroglossum edwardi being sister to the Brevicipilla group instead of the entire genus like we saw in the stacks tree. So there are some points where the topology changes, specifically for Porroglossum edwardi, Atrix, and Rodrigoi, which I already touched on. And then the species Equigeneraceae in one tree comes out, I believe, sister to the entire genus, but it's extremely low support. It's one of the trees that had, I think, a 75% similarity threshold, so a lot of data was thrown out. So didn't have enough resolving power to figure out exactly where it should be on the tree. So essentially moving on from now on, this is the tree that we're utilizing, the pyrite tree. And the reason why we chose pyrite over stacks after all of the parameter testing is based on how both programs were described in the paper. For instance, pyrite is utilized to incorporate indels, whereas stacks, if it finds an indel, it just splits it and counts it as 2 loci. Pyrite will be able to cluster it together and consider it one locus, so you're going to have a lot fewer loci, but loci that are closer to true loci. Also, it incorporates reverse complement clustering. So when you use genotype by sequencing, sometimes it'll flip the sequence one way or another, and if it's reading trying to cluster them together and it's turned the wrong way, then it'll consider 2 separate sequences and stacks, whereas pyrite can put them together. So you're going to have a lot less erroneous loci with pyrite. And it was, of course, built for phylogenetic inference in the beginning of the Orchidaceae pyrite. So, yeah. For the principal component analyses, remember I looked at flower shape, labellum shape, and petal shape. I didn't show petal shape or labellum shape here because there was a lot of overlap, seemed to be a lot of convergence, which it seems to be some convergence here as well for flower shape. But it does look promising, at least from the data that we were able to gather. For instance, here you can see the Porroglossum group starting to separate from the Brevicephalin and kidney groups. Again, here as well, part of the kidney group is nested within there. And then we can see separation there as well. So for overall floral shape, it seems that we do get separation within the groups. However, there is still convergence as well. I'll get to that in the conclusion more about what we can do to improve that. And then I looked at evolutionary shifts with L1OU, like I told you, for all of the morphometric traits with all of the trees as well. Here with the pyrite tree, this was the only result that actually gave us something meaningful. So for the overall floral shape, you can see that even though it's low bootstrap support, we show an evolutionary shift for that Brevicephala group in terms of overall floral shape. So it appears that morphometrics could be useful in diagnosing certain groups of Porroglossum. However, more work needs to be done in order to determine if these characters are powerful enough to as synapomorphs. So chapter 2 conclusions. Extensive parameter testing appears to help minimize the effect of paralogs and sequencing error in the final data set. However, a holistic account should be accounted when one looks at the number of loci and SNPs recovered, the error rates for the loci, SNPs, and alleles, as well as tree topology. So taking into account all of those at one time in order to figure out what the best parameters are to go with. Again, remember I said it's kind of subjective. However, you have a lot of different avenues to look at and compare and contrast to see which ones make the most sense biologically in terms of tree topology. Then for GBS loci, they fully resolve phylogeny and Porroglossum through the stacks and pyrite pipelines, as I showed you. We had 3 monophyletic groups consistently recovered: Porroglossum, Echidnae, and Brevicephala. However, we had those 4 species bounce around the tree: Edwardi, Actrix rodrigoi, and Equigenerense, which could be due to some type of biological process, or it could be due to just a lack of data in some of the data sets where we have a higher clustering threshold, like 75%. Again, I told you we stuck with the pyrite pipeline moving forward, and the reasons why. Again, morphometrics seems promising in relation to overall floral shape. However, petal and labellum shape showed a lot of convergence, which we would probably expect in terms of pollinators' size and different things like that. However, we think that maybe adding more quantitative measures, such as looking at the length and width of different traits, such as sepals, petals, etc., may help to create wider separation in those PCs to be able to have synapomorphies to diagnose these groups. And then the pyrite phylogeny revealed a shift for the Brevicephala group in terms of overall flower shape, but not in any of, none of the other traits showed any shifts for any of the groups. All right, so chapter 3 is the kinematics chapter, introduction. So Charles Darwin, we all know, wrote on the origin of species, right? So his theory of evolution by natural selection. However, what's sometimes lesser known is that he published 3 additional books afterwards, in addition to many more, right? I'm just highlighting these 3 to provide support for his theory of evolution by natural selection. On the movements and habits of climbing plants, power movement in plants, and insectivorous plants. We looked at things such as the snap tentacles of Drosera or sundews, and also the Venus flytrap. So we can essentially consider Charles Darwin as the pioneer on plant movement, right? He's one of the first people to actually sit down and study these aspects. And in much more recent times, in the last few decades, we see a lot more studies that are being published on plant movement, as well as animal movement as well. So I'm only highlighting 2 plant movement studies here, but just quickly I'll tell you of another study in animals where they look at mantis shrimp and how fast the claws strike for those. So that would be an example of a kinematics study that was done with animals. With these 2 studies here in plants, we have one with the Australian sundew, where they looked at how fast the snap tentacles snap, and then also one on Catasetum, which Darwin considered the most remarkable of all orchids in one of his books, on fertilization of orchids. And in this case, they determined how fast the male flowers eject the pollinia. However, I think that if Darwin had come across Porroglossum, he would consider Porroglossum equally as interesting. Maybe it would be the most remarkable of all orchid genera. So here you can see another example of the floral pollination trap in the open position and then the closed position. Here's some Drosophila type flies possibly, or maybe just Diptera. I'm not exactly sure what type of Diptera, I shouldn't just say that, but some unknown Dipteran there on the plant. But essentially an insect lands on the labellum or lip and then it snaps closed, and then you have the column, the column with the pollinia that would attach to the insect, and then it would presumably go on to the next flower for pollination to occur. So I'm going to show you a few videos that I took in Ecuador. So like I said, a lot of my work was looking at the snap trap and how it varies. So this is the Brevicephala group here, the one that was in blue earlier. You can see that it has a very quick snap trap, within a few seconds. The Echidnae group are the ones that have the hairy peduncles. Again, it has a pretty quick snap trap, fairly quick. However, when we look at the Porroglossum group, remember that non-recipinate group, the time that it takes for the snap trap to close drastically increases over a minute and a half. So you can see at time 0 we apply a stimulus, and then it slowly closes over time. You can see here that it's further along, and then it will rapidly snap shut at the end, as you'll see with some kinematics plots that I have later on. So research questions: is there interspecific and intraspecific variation in terms of scalar and vector quantities? By scalar, I mean snap trap closure time, how fast it takes for the snap trap to close. Vector quantities, I'm talking about displacement or direction, speed with the direction, right? So how fast it's moving in a certain direction, in this case vertically or upwards. And then average snap trap acceleration. Do closely related species of Porroglossum resemble one another more than expected in relation to previously mentioned quantities, or in other words, is there phylogenetic signal for these traits? Do these scale and vector quantities produce evolutionary shifts through that package L10U with Ornstein-Uhlenbeck models that I told you about that I use for morphometrics? And then is there a correlation between snap trap closure time and the elevational occurrence of these species? Or where the species are found in situ? All right, so chapter 3, material and methods. This is my study site in Valleceo. So like I said, I did a lot of my work in the greenhouses. So pretty much this entire bench was mine for a couple of weeks. And I used camcorder to record the snap trap, just like I showed you, applying a stimulus to the trap and recording until the snap trap was fully closed. So the very first time I went to Ecuador in 2014, I didn't realize that I could calculate velocity, so I didn't actually include a scale. So a lot of those first videos, which I think it's only a subset of a couple hundred videos, didn't incorporate distance. However, I had over 900 videos, as you can see here. So many of the videos did incorporate distance, so I could calculate velocity and acceleration. Okay? So the study site, again, was in Ecuador at the Orchid Nursery Ecuagenera. Collected 914 videos spanning 33 species for snap trap closure time, and then only 31 species for velocity and acceleration, the ones that were lacking the tape measure scale. So we did high-speed videography at either 30 frames per second or 60 frames per second. 30 frames per second being the first time we went to 60 frames per second with a better camera. And then I used the tripod every time to minimize motion by hand blur, so essentially to keep the camera still so that it would only be recording the flower closing and not have any background stimuli. And then I applied a mechanical stimulus with a bamboo skewer each time. Calibration scale, I just used a tape measure where I placed it directly at where the flower was so that it was proportionally scaled so that I can measure the distance that the labellum traveled, and then I can calculate things like acceleration and velocity. So then I used this program called Tracker, which was made by a physicist named Dan. So it's really cool because it allows you to track the movement of anything through time. So if you have a video of something and you put a mass point on it and you start tracking it through time, it will create a graph and show you its velocity if you have a scale in the video as well. So then I calculated these vector quantities, change in S over change in T, which is just change in displacement or distance over time, right? And then change in velocity over change in time for acceleration. I ran a Shapiro-Wilk test for all of those traits to determine if they should be logarithmically transformed. And in all cases, I found that yes, they should be logarithmically transformed. Then I used R packages to map the character traits onto that pyrite phylogeny that I showed you, measure evolutionary shifts on that pyrite phylogeny, and then lastly, I utilized linear regression analyses to see if there was a correlation between snap trap closure time and elevational data. So chapter 3 results. Here we have a logarithmic box plot column graph of snap trap velocity on the y-axis, and then all of the species of Porroglossum here on the x-axis, and their names here with the corresponding groups. So here you can see all of these species from the Brevicipilla group have really fast snap traps, right? So really high velocity, distance over time, really fast snap traps. Those are the ones with the dorsal sepal that bends backwards, if you recall. You have the Edwardi group, right? The monotypic Porroglossum edwardi, which has a lot slower snap trap, even though we found it either sister to the entire genus or sister to the Brevicipilla group. So a change there. The Echidnae group, the ones with the hairy peduncles, also have very fast snap traps comparable to the Brevicipilla group. Lastly, that Porroglossum group with the non-resupinate or resupinate flowers. Most of those species conform to really slow snap traps. However, we do have two species that are kind of outliers. They conform to a much faster snap trap like the Brevicipilla and Echidna groups, which I'll get into a little bit later of why that might be the case. Right, so I also did this for snap trap closure time and also average acceleration, but in order to not bore you to death and show you the same trend over and over, hopefully you'll just trust me that the trends were very similar to what you're seeing here. So this is just a tree that I mapped the continuous trait of average velocity onto using phi tools, right? So cool colors represent slow snap traps and warm colors or dark reds represent fast snap traps. So again, here you can see for the Brevicipilla group, we have fast snap traps, right, that are red or orangish. For the Porroglossum group, we have mostly slow snap traps represented by a blue color, except for Lycaenum and McDaniel, those two that were conforming onto the Echidna and Brevicipilla trait. And then the Echidnae group here with mostly fast snap traps as well. So just another way to visualize which species have faster snap traps and which ones have slow snap traps. And I didn't show it here, but I also ran phylogenetic signal for Pagel's lambda and Blomberg's K statistic. And for all of those continuous traits, I found that there was very high phylogenetic signal. In all cases, highly over 1 and p-values much less than 0.01. So indicating very high phylogenetic signal, or that species resemble one another more than chosen at random on the tree. Okay, so this is average velocity, looking at evolutionary shifts under Ornstein-Uhlenbeck models. So for this case of average velocity and average acceleration, we got the same trend where we had 3 different shifts. However, for snap trap closure, we didn't recover this shift, but we'll just focus on this one for now. As you can see, the Porroglossum group, the ones with the non-resupinate resupinate flowers, represents a shift to slow snap traps with 86% support. Then a shift back to faster snap traps for Lycaenum and McDaniel, those outliers. And also a shift to a slower snap trap for Porroglossum edwardi in comparison to the fast snap traps of Echidna and Brevicipilla. And then these are just the bar graph values for the continuous trait average velocity. Okay. And this is just also a shout out to Cecile and also Ricardo that helped develop this package for L10U. Okay. So then the next few figures going forward are looking at displacement versus time graphs, or in other words, distance with a direction. So remember I said I'd use that program tracker and track the libellum through time. So imagine that the libellum was right there and I put a pointer there and I continue to track it, it would give me a graph, right? As long as I have a scale for distance. So here you can see once the stimulus is applied for Porroglossum olivaceum, it's one from the Brevicipilla group. Within a matter of 2 seconds, we have an exponential closure of the snap trap. So really rapid closure like I showed you in that video. For Porroglossum edwardi, we also have a really rapid closure of the snap trap, but it takes a lot longer for that to occur. You can see that there's a long period of essentially dormancy before it rapidly closes. For Porroglossum apolloe, or the Echidnae group, it conforms to the Brevicipilla group. They're very similar. Within a few seconds, we have an exponential closure of the snap trap as well. And then here's the biggest difference with Porroglossum, with the Porroglossum group, where oftentimes over a minute, 2 minutes, even 3 minutes is how long it takes for the snap trap to close. And in a lot of cases, it has this little dip in the graph here, where essentially the labellum will slowly move into place, and then it'll have a dormancy period and then snap shut. So it could be some type of way to conserve energy by having to snap a shorter distance or something like that. However, you can see that it takes a lot longer for the snap trap to close. But once it does close, it's an exponential closure, just like the rest of the groups. And then this is just a figure that shows all 4 of the Porroglossum groups on one graph. So again, the Porroglossum group versus Edwardii, which is kind of intermediate between fast snap traps here on the left and slow snap traps here on the right. And then this is one of my linear regression analyses looking at snap trap closure time on a logarithmic scale in relation to elevation. And here you can see we have a negative correlation, or a negative correlation coefficient. I didn't put it here, but it's around negative 0.7, which is fairly high, showing that we have an inverse relationship, or in other words, species that have really slow snap traps occur at really low elevations, and species that have much faster snap traps occur at much higher elevations. However, we have some of those outliers like Lycaenum. Remember, that was one that was in the Porroglossum group that were slow, however, it's a little bit faster. Here we can see a reason why it might be moving faster. It's occurring at a much higher elevation than the rest of the species within its group. So it's essentially maybe occupying a different niche or habitat in that case. So chapter 3 conclusions. For continuous trait data, in other words, time, velocity, and acceleration, I found high levels of phylogenetic signal for all 3 of those, suggesting that species resemble one another. Fast action snap traps appear to have evolved twice in the Brevicipilla group and the Echidnae group. Multiple evolutionary shifts in trait evolution were detected using L1OU. So we had a shift to slow snap traps for the Porroglossum group, the non-resupinate and resupinate flowers. A shift to fast snap traps for 2 species within that group, Lycaenum and McDaniel. And then a shift to a slow snap trap for 1 species within Brevicipilla group, or sister to the Brevicipilla group, Porroglossum edwardii. And then there appears to also be a correlation between time of snap trap closure and elevation. For instance, Porroglossum Lycaenum is unique. It has a moderate snap trap and occurs at around 2,100 meters in comparison to the other species that occur at a lot lower elevation. One of the hypotheses that we have is that there are a lot less resources available at higher elevations. There's been a few studies that have been published that show that when the temperature is decreased, fecundity goes down, so there could just be less insects that are flying around at higher elevations. So by having a faster snap trap, you're essentially making it count when an insect lands on you for pollination, because it might be your only chance compared to a flower that's at much lower elevation where it's warmer and there's a lot more resources, a lot more flies flying around. And then chapter 4, like I said, is very short. It's not anywhere near the other ones, but I'm just going to show you essentially my results here, and I'm not going to go through all of these organic compounds or anything like that. I just wanted to show you something really unique where almost all of the species, minus Porroglossum tripolex, seem to have a very similar fragrance profile. However, when you smell them, you would notice that a lot of them have different scents. Some of them smell very fruity or floral. Some of them smell like what I characterize as a wet dog. And then some of them are others are also really foul as well. Even though they produce compounds that are relatively the same, minus Porroglossum tripolex. However, if you've ever looked at Robert Gusso's paper, How to Make a Scent, he proposes that there's over 5 ways to make a scent. And one of those ways to make a scent is, instead of changing the types of compounds that you utilize to make your scent, you just express them in different amounts. So if you have the same amount of compounds, but you express them at a higher amount or lower amount, you should be able to make a different scent, and it would be more energy efficient to do that versus trying to incorporate an entire new scent. So preliminarily, at least, it seems that species of Porroglossum are starting to do this where they're utilizing the same amount of compounds at higher and lower ratios than others. And then this is just my dedication slide that was in my dissertation. So my grandmother passed away last fall, and my great aunt passed away a few years ago. When I was young, my dad was working a lot, so they were pretty much instrumental in making me the person I am today, and always encouraging me to pursue my dreams. I remember when Ken sent me the email that I was accepted into graduate school, my dad was like, "Oh no, you're not going to grad school. Don't do that. You're gonna go up there and horrible things are gonna happen and you're gonna be on your own." And my grandma was the only voice of reason. She said, "Boy, go follow your dreams and don't let anybody discourage you." So I gotta thank those two for getting me where I am today. So I definitely have to put that in for them. And then this is my acknowledgement slide, of course, to my committee. I hope to have a meaningful discussion with y'all after this and how to move forward with publishing my chapters. Coding friends, Steve Goldstein's been really instrumental in helping me learn a lot of the commands to do all of those GBS analyses. Ricardo's been huge as well with learning R. I know he says a lot that I learned most of it, but he was really great in teaching me the basics and sending me a lot of code that I was able to manipulate and modify to fit my needs for my data. Daniel Spaulding really helped me get on my feet when I first started utilizing GBS data, and I had no idea how to even change a directory on the computer. And then, of course, Chloe and Rachel and I spent many hours basically banging our heads against the desk trying to get things to work, and going over to CHTC many times. So they helped me out a lot as well. And then, of course, my various funding sources, NSF, AOF, and then the departmental awards that I got, such as the Davis Grant and ILTIS Biodiversity Grant. I think that's it.